So, hi, hi everybody. Thanks so much for joining us today for another webinar from the International Wheat Genomics Consortium that we are organizing. It's my pleasure to have today Dr. Julia Kreiner here with us leading our discussion today. So a brief introduction about Julia. So she's a new assistant professor at the University of Chicago in the Department of Ecology and Evolution, whose lab studies the rate and nature of a plant adaptation to contemporary change. Her work has focused on testing these concepts in the rapidly evolving weeds, Amaranthus tuberculatus, or common waterhemp, whose range in North America predates the agriculturalization of the landscape. Integrating population genomics across space and time, her work has unraveled the relative importance of new mutations, extending genetic variation, and gene flow in the spread of herbicide resistance, phenotypic and genomic signatures of adaptation to agriculture, and the tempo of these evolutionary processes over the past 2 centuries. So Julia, it's my pleasure to have you here today. I'm super excited to see your presentation. Just a quick reminder to our attendees, if you have any questions, please enter them in the Q&A box here at the bottom of your screen, and then we're going to discuss your questions later on. So Julia, take it away. Thank you so much for accepting again our invitation, and yeah, it's a pleasure to have you here today.
Thanks, Kaio. Yeah, really grateful to be able to join you here today for the International Wheat Genomics Consortium webinar series. And I'm going to tell you about some work I've been doing and then my group is looking to do to understand the origins and spatial temporal signatures of selection on resistance mutations. And so I'm just going to have a little plug here that my group just started at the University of Chicago, as Kaio mentioned, and if any of these concepts or work interests you, please don't hesitate to get in touch, as my group is recruiting at all levels. Okay. So I think we're all aware of the global impact of agriculture, and in large part, this is out of necessity, right? By 2018, our world's population had already surpassed 7 million people, or 7 billion people, I should say. And so to feed this world, the world's global population, we've used over 50% of Earth's habitable land towards agriculture. So of course, this has profound benefits for public health, but it also has important impacts on our natural resources. And what I want to focus on today is how it's really a key driver of change. And so that change is not just in terms of land management, but also the evolution of organisms across these landscapes. So we can visualize kind of this landscape change another way, and that's by looking at crop land use over the last 4 centuries. So at its minimum in 1600s across Canada and the United States, we were using 1 million hectares of land towards food production. But there was exponential increase since then, and at its maximum in the 1950s, we were having over 500 million hectares, or 10% of Canada and United States land surface. And so this change, or this period of agricultural expansion, was really driven by cultural necessity and massive population growth that occurred during this time period. And so at the start of the Industrial Revolution in the 1800s, we actually took more than 50% of North America's workforce out of agriculture and into other industry. And this kind of was this period of expansion and industrialization of agriculture. And it wasn't really until the 1950s where we realized, there's a finite limit in how much land we can convert to agriculture to increase crop yields. And so during this time, there was really a shift from expansion to the intensification or increased efficacy of agricultural practices. And I think it's exciting to recognize is that this really started with an evolutionary innovation, and that was the breeding of high yield crop varieties. And so along with that, though, came highly controlled irrigation, widespread mechanization, and increased, and some would say, over-reliance on chemical inputs like pesticides, fertilizers, and herbicides, right? And so this increased efficacy of agriculture has, I think, really not only had an important impact on increasing crop yields, but also intensifying selective pressures across these landscapes. And I think this is really typified by the rapid and unintended response of herbicide resistance evolving across species, right? And so we know that this is rapid because herbicides were only introduced across the landscape in the 1950s, and the first case evolved just 6 years later. Since then, resistance to herbicide has arisen across hundreds of species in response to almost every chemical mode of herbicides. So this is a huge limiter of how efficacious our agricultural practices are. Indeed, while this estimate's outdated, we've estimated about $33 billion of costs of herbicide resistance by drawing down on average about 12% of crop yields in the United States alone. Right. So because of its importance, there's been a lot of work done to understand not only the gene targeted by these herbicides, but the particular amino acids and non-synonymous mutations within them that confer resistance to herbicides. So this sets up a scenario that, it's really pressing actually for understanding how resistance evolves, how often, how quickly, but we're also in a situation where we now have the technology to address these questions, especially in this species, Amaranthus tuberculatus. So this is a species that has thrived despite the conversion of its native habitat to agriculture, right? So it's a native plant, native to the Midwestern United States, but over this time period of especially the intensification of agriculture, it's become one of the most problematic weeds in North America. In large part, that's due to widespread herbicide resistance across these species, across the species. So individuals can harbor cross resistance to every chemical compound of herbicide. But beyond that, there's some interesting ecological features of the species. So it's dioecious, meaning it has separate sexes. It's wind pollinated, it's diploid, but it also has genetic diversity structured across the range. So there's 2 native subspecies that were historically isolated on either side of the Mississippi River. And together, these features really present a system where there might be lots of potential for widespread genetic diversity and gene flow facilitating rapid adaptation. So we've been testing some of these questions, how this aligns with land use and herbicide practices through time, and developing this system from a genomics framework as well. And I really want to highlight my collaborators on this work, especially Patrick Trinnell, the University of Illinois, who's been developing Amaranthus and waterhemp specifically as a system for weed population genomics for several decades. My PhD advisors at University of Toronto, Stephen Wright and John Stinchcombe, who supported me through this work. Detlef Weigel, who contributed to some of the first genomic resources in the species, and my collaborator on some of the work I'm going to be talking about today, Sally Otto at the University of British Columbia. So today I'm going to highlight how we can test some of these concepts about the rate at which adaptation to agriculture and herbicides has evolved in water hemp, and how origins of herbicide resistance might contribute to that rate in 2 different parts. So for the first one, we'll just get right into it, the strength and tempo of adaptation to agriculture. I think it's important that we start with kind of this conceptual framework for understanding what determines the tempo at which populations can actually adapt to environmental change. So when we go in and we quantify how rapidly Amaranthus has evolved, this way we have a context for understanding, well, how generalized are the patterns that we see, and what mechanisms might facilitate that. So one place to turn to is actually a theory at the intersect of the fields of ecology and evolution, and that's evolutionary rescue theory. And so this theory posits that it's really the interplay between ecology and evolution, or population demography, and the availability of adaptive genetic variation that determines whether or not a population is likely to go extinct or undergo adaptation or evolutionary rescue in the face of environmental change. So we can imagine that some change here occurs at time 0 on this x-axis. I wonder if you can see my mouse. Hopefully you can. And that could be okay, great. That could be the application of herbicides. And so imagine herbicides are applied. These are designed to kill a large proportion of the population. And here that's successful. We see this population demographic decline. But whether or not this population goes local extinction or extirpation, or undergoes local adaptation depends on whether or not there's sufficient adaptive genetic variation. So in this case, this population actually had a new beneficial mutation arise. That might be a large effect target site resistance mutation that allows this rebound of the population size and for adaptation to occur. And so while this is bad news for farmers, it's good news for the population. They've actually persisted. But it's important to take a look at these dynamics. And what you'll notice is there's actually quite a long waiting time for these new beneficial mutations to occur, right? And so in this way, we think of adaptation from these new mutations to be a slow process. Whereas in larger populations where there might be more individuals across the landscape, there might be more mutations segregating across them, we expect higher levels of standing genetic variation that might contribute to a much faster rate of adaptation. So there's already a mutation that was segregating in that population that might have been rare, that is now favored in these new environments. And so that actually prevents much population size change and allows adaptation to occur very rapidly. And so indeed, the role of standing genetic variation in rapid adaptation has been increasingly recognized in numerous different systems, and can help us kind of contextualize or develop expectations for the rate of adaptation to contemporary environmental change. And so with this context in mind, I was very interested in characterizing, well, what does agricultural adaptation look like in water hemp? What phenotypic and genomic changes have had to occur in order for this plant to become invasive in these agricultural settings, right? And so one really exciting component of the system is, remember I told you that this plant's actually native to agriculture, and actually populations still exist in kind of this ancestral habitats across the range, that is these natural habitats. And so we performed pairwise sampling of agricultural and natural and agricultural habitats across the range. And just give you an idea of what these look like. So here's a set of these habitat pairs that we collected just less than 3 kilometers apart here in southern Illinois. This natural habitat is kind of typified by a pretty sparse population of water hemp. You can see it here in this corner. Pretty small individuals occurring on the sandy beach, not much competition, pretty dry habitat, although some individuals here were occurring right actually on the edge of the water. But less than 3 kilometers away, a soy field, just a couple weeks out from harvest, completely overrun by water hemp, many meters tall. So very different ecological and environmental factors across these different habitats that we expect to shape differences in fitness across them. And that because we have this repeated pairwise sampling, we can really look to disentangle broad scale drivers of phenotypic and genetic variation across the range from consistent differences across these two habitats that have shaped adaptation to agriculture. Okay, so the first thing we did was actually grow out plants from these seed collections that we made. And we did this in a manipulative common garden experiment when I was finishing up my PhD actually at the University of Toronto. So you can see thousands of pots here. We actually grew out 6,000 water hemp individuals in 3 treatments. So that's why I call this a manipulative common garden experiment, because we wanted to recapitulate important parts of both these natural and agricultural habitats. So we grew out plants in a water supplemented treatment. This is to recapitulate an important part of these natural riparian habitats. We also grew out plants in agricultural habitats or in competition with soy to represent an important part of these agricultural habitats and in a control treatment. And why we did this is to kind of pay homage to a very classic experiment that is kind of the gold star approach for identifying local adaptation. And that's a reciprocal transplant experiment. And so what you would expect to find if there's local adaptation is that the fitness of plants sourced from agricultural habitats would be higher when they're grown in soy than plants that were sourced from natural habitats. And in contrast, the fitness of plants from natural habitats would be higher than those from agricultural when they're grown in water. Right, so it's a trade-off of this fitness that kind of is is typified by this home site advantage. And so while we looked for this signal, we did so with kind of 12 different traits that we phenotyped across these thousands of plants. And what I'll also focus on today mostly is kind of results from whole genome sequencing of about 200 individuals from these collections. But just to give you some context for, well, what traits have actually evolved during this invasion into agriculture, what I'll just show you is how these individuals, environments, and populations differ in some of these key fitness-related traits across these three treatments we grew them in. And so I'll just color them by what habitat those individuals were sourced from, agricultural or natural. And so we can see there's a ton of variation, genetically determined variation, right? Because all of these plants are grown in the same habitat or the same treatment, but there's a ton of variation in these phenotypes in water hemp. But what we tend to see is that these agricultural plants tend to be larger, to have more biomass, regardless of the treatment that they're grown in. But they also have a longer days to flowering, right? So we know there's been a clear shift in the phenotypic optimum or genetic diversity that underlies these traits because they've been grown in the same habitat. So this is genetically determined differences that are consistent across the range. But we're seeing pretty limited signals of local adaptation because, agricultural plants, while they tend to, have higher biomass in grown in soy, there isn't that crossing over. They don't have lower biomass when grown in water. So these agricultural plants are doing pretty well regardless of where they're grown. But there's lots of shifts that have had to occur during this species invasion into agriculture. So another way we look to kind of decipher the signature of agricultural adaptation is through genomic approaches. And that's what I'm going to focus on kind of for the rest of the talk. So from these collections, we sequenced 182 individuals. And we were able to get information for about 10 million SNPs across the genome. So lots of data in order to test kind of for these signals of agricultural adaptation. But the first thing we wanted to do is, considering all of this data as a whole, how different are natural and agricultural populations across the genome? And so when we ask whether agricultural plants have genetic variation that are unique to them relative to natural plants or populations, the answer is actually no. So FST, which describes that exact question, it actually is approaching zero. So all of the variation we see in agricultural plants tends to be shared on average across the genome with nearby natural populations. And so this is important in and of itself because it tells us that the practices that we implement in agricultural environments are really important for shaping genetic variation in nearby natural habitats. But you might ask, is that even the case for resistance mutations? Well, what's exciting is that we can assay the frequency of resistance mutations across these two habitats in a pretty systematic fashion, right? Because we have population pairs that we'd expect natural populations to have much lower frequencies of resistance than agricultural ones. And so for 6 common resistance mutations, we looked at it exactly that. We plotted the frequency of these causal resistance mutations. So you might recognize some of these, that deletion within PPO, the amplification of EPSPS, mutations within the amino acid position 376, 574, 653 of ALS. And what you'll notice is that some of these mutations, there absolutely is constraint on the frequency of these mutations in natural populations. And so when we connect paired agricultural and natural populations, we tend to see that while agricultural populations might have very high frequencies of resistance, their nearby natural pair is constrained to be at much lower frequencies. And so this is strong evidence for, despite migration, introducing these mutations into these natural populations, a cost of these mutations that constrains them to lower frequencies. However, you'll notice that's not the case for many of these resistance mutations. So for the amplification, we're actually seeing some pretty high frequencies of resistance in nearby natural populations, and also for these very common ALS mutations, quite the same. And so while looking at these frequencies of resistance across habitats can shed light on the cost of these mutations, I think what it's also showing is these costs are extremely variable across these resistance mutations. And this paper has done some work to quantify what the cost to benefit of these mutations are across different habitats that I won't go into too much today. But with that in mind, you might be curious, okay, well, this, for example, this PPO deletion seems to show strong differences in their kind of fitness across habitats. Are there other mutations across the genome that show kind of this signal as strong? And so we can systematically test for a signal of agricultural adaptation at every site in the genome. In particular, we can ask whether consistently across these pairs, if there are SNPs that are overrepresented in one environment compared to the other. And so a kind of generalized form of that test is called the Cochrane-Mantel-Haenszel test. And on the y-axis now, I'm just plotting the significance of the effect of the SNP for agricultural adaptation, right? And so this is just a classic Manhattan plot, just illustrating the strength of this effective agricultural adaptation at all sites across the genome over 16 chromosomes. And I'm highlighting in red that about 500 SNPs across the genome show strong evidence of playing a role in agricultural adaptation. These are consistently differentiated across habitats. And together, these alleles are enriched for functions in terms of growth, reproduction, drought, stress, and of course, herbicides. And actually, when we look in what region of the genome shows the strongest signature of agricultural adaptation, it's actually here on chromosome 11. And this peak actually falls directly within the gene PPO, the target of PPO inhibiting herbicides. So that figure here that I showed you depicting the difference in allele frequencies across natural and agricultural habitats is actually the strongest difference, consistent difference in allele frequency we see, really kind of emphasizing the importance of herbicides in shaping adaptation to agriculture. I think an exciting part, in addition to producing a ton of hypotheses about what genes, what functions might be involved in adaptation to agriculture, is this suite of agriculturally adaptive alleles also provides us a framework for testing what processes and what historical processes have contributed to the patterns that we're seeing at these alleles, their frequency across populations over contemporary landscapes. In particular, we might be, you might be, if I'm giving this talk, well, thinking back to evolutionary rescue theory and interested in how demographic change, but also selective shifts might have shaped these patterns. And so what's really interesting is that we can look directly and observe their evolutionary history directly through time in herbarium samples. So this is a time series that has been preserved in plant museums over the time scale at which agricultural shifts have occurred and herbicides have been introduced. So working with 8 herbaria across the Midwest, I was able to curate a set of about 108 samples that span the same scale of collections, but dated back to 1828. And these specimens have been so carefully annotated that we could also characterize whether or not they were collected from natural, agricultural, or disturbed habitats. And so in this way, we can not only ask how the frequency of these alleles have changed through time, but how that's been shaped by changing selective pressures across these different habitat types. Okay, I'm gonna first start with demographic change and how that's contributed to the patterns that we're seeing at these loci across populations. And in working in waterhemp, I'd be remiss if I didn't mention or highlight the work that Jonathan Sawyer did based on these same herbarium specimens in waterhemp. So in 1956, he characterized phenotypic diversity in these specimens, and he hypothesized that it was actually this expansion of the southwestern Varudis variety northeastwards that had led to much of the agricultural invasion that we're experiencing today. So he himself was thinking about the role of standing genetic variation in adaptation to agriculture. And I think what's really exciting is that we can test these hypotheses now directly with our herbarium data. Oh, I skipped ahead a bit, but this is also to show that, like, compared to Rudis, this variety tuberculosis' range largely overlaps with the major Mississippi and Missouri water basins of North America. And so it's very much typified by these natural riparian habitats in contrast to this more ruderal weedy species. So this contributed to some of Sawyer's hypothesis about adaptation of Varroa to agriculture. Okay, so from our herbarium specimens and our contemporary data, we can estimate how much of each individual's genome comes from Varroa ancestry. And what I'm going to show you here is how that is distributed across space, but also through time. And so like I explained, Varroa ancestry is very prevalent in the Southwest. Varroa is very prevalent in the Northeast. But what you'll notice here is there's been a shift in the distribution of ancestry, that on more recent timescales, Varroa has actually shifted eastwards. And we can observe this because we have time series data through time. So this shift in the distribution of ancestry. But if we color each individual, not by what time frame they were collected from, but what environment they were found in, we can then see that while historically there was no difference in the distribution of ancestry across agricultural and natural habitats, that this shift through time has actually driven the sorting or the excess of Varroa ancestry into agricultural environments. So this is pretty exciting in terms of testing Sawyer's hypothesis, but we can go one step further and not just ask about these signals across the genome, but how this relates to these agriculturally adaptive alleles that we've identified across the genome. Right? Remember, I showed you that Manhattan plot. If we look at how the strength of these p-values corresponds to differences in ancestry across the genome, what we actually see is regions of the genome with the strongest effect of playing a role in agricultural adaptation are those that have an excess of Varroa ancestry in agricultural compared to natural habitats. So this is really telling us that Sawyer was right, that Varroa ancestry has played a really important role in adaptation to agriculture by providing the standing genetic variation on which agricultural selection has acted. And now, I think with that understanding, we can contextualize much better how that relates to how quickly these alleles across the genome, these agriculturally adaptive alleles, and particular resistance mutations have changed through time. And how we can do this is, again, leveraging our herbarium samples. We can look at a particular site across the genome. So this is just a toy example, but we can imagine that at a particular site in the genome, we can characterize whether an individual is homozygous for the reference allele, heterozygous, or homozygous alternate. And because we know when they are collected, we can kind of visualize how the frequency of these alleles have changed through time, right? And so quantitatively, we can think about a very general model of selection. So for those of you who are mathematically minded, neither am I, but we can really think about how what this model of selection is trying to do is estimate the frequency of some allele, P. You can imagine that as a resistance allele at some time t. So what was the frequency of that allele in the 1950s when herbicides were introduced, for example? Well, we can actually quantify or estimate that if we know the frequency of this allele sometime in the past. Here at time 0, we can ask how many years have changed since we've had that estimate. And we can say, well, if we know the rate change parameter, we can actually solve for that value. And what's exciting is that we can rearrange this equation to take the form of a much more general model, and that's just a logistic regression model. And so what that allows us to do is intuit what happens when we regress these genotypes on collection year. Well, we can actually estimate this P at T, the frequency of some allele at time T. That's just our predicted value. We can extract kind of the slope of this line of the logistic regression. That's going to be our rate change parameter, the strength of selection. Right. And so in this way, we can directly quantify the strength of selection on resistance alleles through time. So now I'm going to show you that for our real data. I'm just going to show you that for 154 of these agriculturally adaptive loci across the genome. So these are alleles that play a very diverse role in functions, right? Growth, reproduction. They might be involved in that difference in flowering time. They might be involved in polygenic responses to herbicides. When we look at the trajectory of these alleles through time, let's first do that for samples from natural habitats. So these are ones that we don't expect to be experiencing direct selection from agriculture or agricultural management practices. The trajectory of these alleles, here I'm just coloring by how much they've increased through time. So in dark purple, they've increased a lot. In yellow, some of them have actually decreased. We see that on average over the last 150 years, these alleles have increased by 6% on average. But over this time frame, it's actually not consistent with any significant signature of selection. So there's no evidence for selection on these alleles over this time frame in natural habitats. But in contrast, when we look at how these alleles have changed in samples collected from agricultural habitats over the last 150 years, they've increased by 22% on average, and in experiencing a rate of change, a strength of selection, nearly 2% per year. So this is very strong over these timescales, right? And you can see that just based on how dramatic these allele frequency trajectories are through time. I think this also provides a really nice framework for testing hypotheses about how shifts in land management and agricultural practices, like this shift from the expansion to the intensification, has related, like I outlined at the beginning of my talk, to shifts in selection through time, right? And so we can actually ask, if we were to fit a single rate of allele frequency change to this model compared to multiple rates, is that a better fit to the data that we observed? And that's actually exactly what we see. We see strong support for a shift in selection that happens to fall directly between this period, this transition between expansion and intensification. And so when we fit these logistic regression curves at the environment level, what you can see is there's almost no change in these contemporary agriculturally adaptive alleles prior to the 1950s, despite them being quite common. They're staying at the same frequency for quite a long time. But on recent timescales, since about 1955, these alleles have increased at an even faster rate than we initially estimated, about on average more than 5% per year in agricultural habitats. You can see that in that light gray line, but also they're increasing in nearby natural habitats, right? And I think this is going back to what I initially showed with that FST analysis, that selection in these agricultural habitats have an impact on nearby natural habitats through gene flow. So the practices that we implement in agricultural habitats leads to the spread of these agriculturally adaptive alleles, even in nearby non-agricultural habitats. And maybe what many of you are waiting for, we can actually estimate the strength of selection directly on these resistance alleles. So this is really exciting because, just like in those contemporary samples, we can look at how consistent differences in their frequency across habitats, what that might mean about the costs of these alleles. We can also look, when we average through time and across space, how have herbicide regimes, how rapidly have they shaped the spread of resistance alleles across the landscape? We know that herbicides were introduced right around the 1950s. And so when we fit a model of selection since herbicides were introduced, we're getting a strength of selection corresponding to a 20% rate change on average per year in their frequency across the landscape. And this is averaging across both natural and agricultural habitats. So pretty tremendous, much stronger than is typically seen. This is like rates of selection, to contextualize, that you might see for kind of COVID evolving to different vaccines, right? So this is lethal selective pressure, as you might expect. That strength of selection varies a lot for different herbicides. So in herbicide resistance mutations, so ALS 574, when we estimate the 95% confidence interval of the strength of selection, it might be anywhere between 9% and 35% on average per year. And you might ask, well, why is that the case? Well, this resistance allele in particular is very common across our collections, both natural and agricultural. Its global frequency is about 35%. So we can see a ton of observations of individuals homozygous or heterozygous for this resistance allele. But in our historical samples, we actually see only 2 observations of these resistance alleles. And so to kind of bridge the gap between the very frequent occurrence of these alleles and the contemporary landscape and their historical frequency, there must have been extremely strong rates of allele frequency change and selecting acting on these alleles. And that's exactly what we see. Notably, you'll notice they're at very rare frequencies and pretty much absent at the onset of herbicides. And this implies that these mutations are not standing. These are new mutations on which herbicides has acted, although we're working to expand our sample size to get a sense of how likely this is to be the case across a broader collection. So to wrap up this section, this work has really looked to understand the signatures of agricultural adaptation, but also the strength and rate at which this has occurred along with land use and management change. So we've found that the genomic and phenotypic signatures of adaptation to agriculture are, despite pervasive gene flow across natural and agricultural habitats, polygenic adaptation through growth, reproduction, tolerance to stress, and herbicides. And that historical processes like shifting ancestry across the range has played a really important role on the availability of adaptive genetic variation. And so not just adaptation to herbicides, right? There's all of these other alleles across the genome that tend to play a role in waterhemp's high fitness in agricultural habitats. And those mutations were present at a higher rate in rudous lineages that once it's expanded over the last 100 years has really provided the variation on which agricultural selection has acted. Finally, we see there's been a really rapid response to agricultural selection and herbicide-resistant mutations, or herbicide selection in particular. So this is within decades, we're seeing strength of selection from anywhere from 5 to 20, 33, 35% for particular resistance mutations. And this is much, much stronger than is typically observed in the wild, right? When we contrast natural selection to artificial selection, we're really seeing here the impact that humans can have on reshaping genetic variation across the range. And that's really been driven by the intensification of agricultural practices. All right, I want to finish off by kind of contrasting how these rates might actually be influenced by the origins of herbicide resistance mutations. And so I've talked a bit about how the genetic architecture of adaptation impacts the tempo, right? We can think about polygenic adaptation to agriculture. There's many mutations all across the genome, each of which have a small effect in conferring higher fitness in these habitats. And because of these mutations, each having a small effect, they can actually persist in these populations even when they're not favored. And that means when amaranthus was finally kind of in these intensive agricultural habitats, that this shift, these increase in slight modifications of the frequency of all of these alleles across the genome could allow a very rapid progression to this new trait optimum. And that's the invasive weed it is today. But adaptation might not be able to occur from little changes all across the genome. Sometimes they have to occur through fewer mutations of large effect. We can think about herbicide resistance mutations. And so in that case, because these mutations are such large effect, they might not be able to persist when they're not favored by selection, right? And we think that often herbicide resistance mutations can have a cost. And so in this case, the rate of adaptation or this progression to this new trait optimum is going to be limited by the input of those mutations into the population. And so this was a question for me: how limiting is new mutation really in the spread of resistance and that rate of the change in resistant frequency that we see across the landscape? So to test this question, we can go to a few common resistance mutations across our collections. This is the ALS gene, target of acetylactase synthase inhibiting herbicides. And there's two very common mutations in our collections. That's this non-synonymous mutation at 574 and 653. And actually nearby, there's a PPO gene, target of PPO inhibiting herbicides. And there's a common amino acid deletion that confers resistance. And so we can look at the evolutionary history of these mutations to quantify their origins and understand how that contributes to their spread. And how we can do that is actually leverage the background background variation on which those mutations exist, right? So that we might call that a haplotype, and we can create haplotype trees that depict the evolutionary history of this region of the genome. And so when we do that, and we actually plot the distribution of resistance mutations across these gene trees, what's really striking to see is that for all of these common resistance mutations, there has been multiple origins, right? And we know that because this origin of resistance is more closely related to nearby susceptible individuals than it is to these other origins, right? That's a signature of multiple origins. We can again turn back to kind of this evolutionary ecological theory to understand, well, why might be the case? Julia just told me that mutational origins might actually limit the rate at which resistance or large effect mutations occur. And so we can turn back to this theory and understand, well, what might be happening here? And so one equation that might shed some light on that is this population mutation rate parameter, which is equal to theta, and the rate at which a new mutation enters the population at each site of the genome every generation depends on the effective population size, so how many individuals exist across the landscape for those new mutations to arise on, and the mutation rate, which we know to be very low in most systems. So here, what we see is that the rate of mutation actually depends on the effective population size. That relates to population demography, just like I had talked about. And when we estimate the effective population size of waterhemp through time, what we can see is that while it's been very consistent, about 70,000 over the last million years or so, over very recent timescales of the last 100 years, or even let's say 50 years since herbicide has been introduced, we're seeing a massive expansion in effective population size that has increased from 70,000, this historical NE, to over 80 million. And when we plug that into this equation to understand the mutational input, what we get is a theta greater than 1, which tells us that each site in the genome, every generation, there's one individual across the landscape that harbors a new mutation. So because of how widespread amaranthus is, we're seeing that adaptation is not limited by mutational input at single sites. That's bad news, unfortunately, for the evolution of herbicide resistance. But despite the fact that we see mutational input as not limiting in how resistance spreads across the landscape, gene flow is also playing a very important role, right? And so somewhere in the landscape, these resistance mutations arise, but whether or not that mutation is subject to selection from herbicides, and how widespread and how quickly that gets depends on gene flow, right? And so when we map these different origins of resistance back onto the populations on which those individuals were found, what we can see is that a single origin of resistance, so here there's 2 for 653, I've colored them slightly different colors of green, there's 3 for 574, there's 2 for PPO. When we map those back onto those populations, what we can see is that a single population might harbor multiple origins, multiple distinct origins of the exact same resistance mutation, and that a single resistance mutation, like here in green, is found across multiple populations, multiple agricultural regions, and even across international borders, right? And so clearly it's not just about multiple origins of resistance, but about their spread, contributing to the rapid rate of resistance evolution that we've observed. So putting this all together, in this section, I really focused on kind of the nature of this single gene, large effect resistance mutations in amaranthus. And again, we've seen this feedback between demography and the rate of rapid adaptation, so that it's not limited by mutational input because there's so many individuals on which these new mutations can arise, but also gene flow is really driving the spread of resistance across the landscape that contributes to those extreme rates that we observed in the previous section of this talk. And so together, I think hopefully what I've shown you today is really the interplay of these ecological and evolutionary forces in driving the response to agricultural selection. There's been rapid range expansion and gene flow in Amaranthus tuberculatus that's provided the standing variation on which agricultural selection can act. These allow these populations to persist, right? These are fit enough to persist in these agricultural habitats such that they can experience selection in these new environments and eventually evolve de novo resistance, large effect resistance mutations that further allow these highly fit agricultural populations to expand across the landscape. So I think we're now completing this feedback loop of the kind of rapid continual evolution of Amaranthus to these new environments that we're constantly cultivating them in. And I think this framework is really interesting and important for thinking about the timescales at which we need to be managing resistance evolution and how quickly these processes actually feed back and occur on the correct spatio and temporal scales. So with that, I just want to thank you guys so much for joining today, for listening to the work that we've been doing in the Kreiner lab, now at UChicago. If this work interests you, again, please don't hesitate to reach out. But I also want to thank all my collaborators on this work, Sally Otto, who contributed to the modeling of the trajectories in these herbicide resistance alleles, Stephen Wright and John Stinchcombe at the University of Toronto. And thank you guys again for listening, and I'll take any questions if there are any.
Thanks, Julia. Great talk. Thank you so much. We have quite a few questions here, so I can read the first one.
Okay,
Did you run any GWAS for different herbicide resistance phenotypes to identify genomic loci associated with metabolic resistance?
Yeah, great question. So yes, actually from a different data set, let's see. In this data set, so this was actually in collaboration with Patrick Trinnell. Some of the first work we did was to understand glyphosate resistance in these samples. So I'll show you. And in part, because it had recently expanded into southwestern Ontario, glyphosate resistance was segregating. It was never an issue prior, despite Amaranthus being there. And so Patrick Trinnell's group actually helped us with phenotyping for glyphosate Roundup resistance in these samples. And while we found really widespread EPSPS amplification in these samples, that actually only explained a fraction of the variation in resistance to glyphosate. So if I remember correctly, this explained about 40% of the variation in resistance. So what else is contributing to resistance in these samples? And so I have a molecular ecology paper where we performed genome-wide association, controlling for these large effect EPSPS mutations to identify kind of the polygenic architecture of resistance to Roundup. And yeah, that work is published in molecular ecology if you want to take a look.
Yeah, thanks. There's another question here about the Manhattan plots and some of the hits that you had. They are asking if they might be related to non-target site resistance genes, and if you did any sort of in silico analysis to annotate them.
Yeah, absolutely. That's a great question. So I expect so, right? I mean, I think I made this contrast between these agriculturally adaptive alleles and then these herbicide resistance loci. But for example, this is just a totally unbiased test asking which alleles are most differentiated, have experienced kind of divergent selection across these habitats. And you can see PPO falls out as the most extreme. Presumably there are other, sites in this scan that come out that are involved in this response to herbicides. And so some of these, though, are very interesting, like cryptochrome 2 is involved in circadian rhythm regulation. That could seem to be playing a role in growth and flowering time. And so I expect many of these mutations and hits might actually be very interesting to follow up with candidate gene approaches to identify their functional effects, including potentially polygenic or metabolic resistance. And that's work that my group is looking to pursue in the future. If you have interest in that, I'd be happy to chat more.
And since you're in this slide, there's a question about what's the hit on chromosome 12.
Yeah,
a pile up here.
It's a nice one to look at. The gene was uncharacterized, that it fell within, and even blasting to other species, we didn't get much information on what the putative function of that gene could be. That being said, since this work has been published, we've recently come out, and other groups, like Patrick Chanel has recently come out with better, and the International Weed Genomics Consortium, a much better version of this amaranthus genome. And along with that comes better annotation of these genes, right? So I'm pretty excited to revisit these analyses now, to see whether we can identify other genes that we missed the first time as a result of annotation quality.
There's another question here. So how much impact do you think the increase in fertilizer and other inputs in agri environments had on increasing population size and subsequent increase in the chance of allelic mutations that provide adaptations to ag environments?
Yeah, it's a great question, right? It's like, I think we've been unintentionally domesticating amaranthus in these agricultural habitats. That's like one way I like to think of it. And, we're providing these crops an ideal environment to maximize their yield. But in doing so, we're doing the same for water hemp. So we've been battling that, right? Of course, with weeding approaches, herbicide applications, all these weed management approaches, is to really contrast these selective changes that are favorable for amaranthus to grow in these habitats. And I'm sure that some of these signatures might relate to metabolism of these fertilizer inputs, but some of my work now is actually to use on-farm, working with farmers to have a better sense of what management strategies have been applied, what herbicides, what fertilizers, and by sampling it multiple times throughout the season and going back each year to actually get a sense of how much of this evolution might be driven by a particular herbicide or by fertilizer inputs or so on and so forth. So I think it's an interesting question that we're looking on following up with.
Yeah, that would be super cool. So another question, did you compare seed production female plant between native and agricultural populations? I recall you presented data on dry weight and plant height, maybe, but was curious about seed production.
Yeah, so we did several other phenotypes. We did 12 phenotypes. Take a look at this evolution paper if you're interested. And dry biomass was an easier approach than estimating seed weight, as if you are familiar with amaranthus, here's some photos. About 90% of this plant becomes reproductive biomass by later on in the season. And so it's a huge amount of work to estimate individual seed weight or individual's seed weight, although we did this for about 200 samples. And what we did was, and in this paper, I think as a supplement, we showed that dry biomass correlates very, very strongly with reproductive seed output. And so we felt comfortable kind of using dry biomass as a fitness proxy in these species, although there's limitations to that, of course.
There's another question. Why do you think some mutations are more likely to evolve multiple times than others? Does the frequency of heterozygosity, homozygosity change through time?
Interesting question. Yeah, I mean, I'm sure that you asked if the frequency of heterozygosity changes through time. One way we could think about it as the frequency of admixture changes through time. So, historically, these lineages of Arbutus and tuberculatus, which have different pools of genetic variation, were isolated. But in the recent times, they've been brought together, which means that we might expect there to be higher levels of heterozygosity, standing genetic variation than there was in the past. And I'm pretty sure that's exactly what we've shown here. We've, estimated effective population size through change, which not only relates to mutational input, it also relates to levels of variation across the landscape. But why some mutations have more mutational origins than others? I'm not sure that's the exact way I would phrase it. I think I would say, why do we observe more mutational origins for some than others? I think that the mutational rate at a particular locus, we would expect to be the same, regardless of what mutation you're talking about. It's just that some of those origins will get lost if they're not selected. If these mutations aren't favored, they're going to be lost to us. And with herbicide resistance mutations, there's such strong selection across so many different fields that these mutations are being maintained as a result of the environment in which they're arising. So I think hopefully I've answered that question in its entirety.
Yeah, that makes sense.
Okay.
Have you looked into what factors in the environment of variety rudis promoted genetic variation that prepared it for adaptation to agriculture? Can you use environmental data to predict resistance?
Yeah, super cool question. That's something we're looking at right now. Actually, yeah, like, so both disentangling what are the climatic and environmental differences in the populations where in these populations that have differences in ancestry, and that might relate to kind of the ancestral selective pressures that these lineages experienced. But also, how does that relate to this pre-adaptation to agriculture? Like, what was it that Var rudis had experienced that allowed it, that selected or allowed variation to persist in those populations that were then favored into agriculture? You know, this is called rudis. So it was thought to be ruderal when it was first named. And a lot of that was observations that occurred alongside these disturbed water basins, actually. So maybe it's these highly disturbed, regularly disturbed environments, perhaps that's selected for high levels of phenotypic plasticity in the species. Another thing we've been thinking about is climate adaptation. So this Var rudis type probably has experienced much drier conditions. And in this transition from a riparian to an agricultural plant, you might kind of non-intuitively, it might be important to be able to persist in the absence of high moisture levels. And so that's actually something we have seen agricultural plants do better in these drier habitats. And so we're looking to try and figure out what might have favored this agriculturally adaptive variation in rudis. And I think it's a cool question to ask.
Thanks. We are running out of time. We have so many questions here in the chat, but I'll read one more.
Okay.
And then we should wrap up.
Okay.
Can you put the strength of selection in the context with the strength of selection in domesticated crops?
Mm-hmm. Yeah, that would be a great thing to do. I think, again, that might be the strongest parallel, because in a way, like I just said, we've been unintentionally domesticating amaranthus, right, by growing it in these agricultural selections. It's been artificial selection for weediness imposed by humans. Actually, I'm working on applying some of these exact same approaches to understand domestication selection. So we're leveraging, in collaboration with folks who study domesticated sunflower, but also domesticated cannabis, how rapidly have allele frequency change occurred over the timescale of herbarium specimens the last 200 years and over millennia with archaeological data, which is really cool in sunflower, how rapidly have these changes occurred. So I hope in the future, I will be able to compare what these rates of selection look like for these unintentionally domesticated amaranthus alleles compared to what we see in other species.
That's awesome. Thanks so much, Julia, for your time and for sharing with us your presentation. It's great. It's been great. I really appreciate your time.
Thanks, Caio. Yeah, great to join you all. And feel free to reach out if I didn't have a chance to answer your question.
All right.
Okay.
Thanks, everyone. We'll see you next time.